Hi, everyone. We are still waiting a few minutes to wait for the last participants. Please don't hesitate to use the chat box if you have any trouble. So we are still waiting 1 minute for the last participant. There is still people connecting. And we will introduce the new webinar, Best Practice for Environmental Detection and Monitoring of Airborne Viruses with the Coriolis Air Samplers. So we will start now. My name is Sophie Dubac. I'm the product manager for the Coriolis Air Samplers at Bertin Technology. We will have 1 hour to speak about the best practices for environmental detection and monitoring of airborne viruses with Coriolis Air Sampler. We will start with the agenda of the session. So this morning, I'm with Dr. Martin Serrano-Sanchez, who is field application scientist at Bertin Technologies. We will have 3 parts in this webinar. First thing, we will introduce shortly our Coriolis Air Samplers. Then Martin will introduce you the tips and tricks to collect viruses with our air samplers, and then he will present you case studies of the use of Coriolis for airborne virus detection. Then, depending on the time, we will have a question and answer session. For that, you will need to use the question box. So for your information, all the microphones are muted in order to minimize the interferences. So if you have any trouble or any question, use the chat box and the question box. After the presentation, we will answer to the question. And after the presentation, if we don't add the time to answer to all questions, we will send you all the answers by email. So to start, again, my name is Sophie. I'm the product manager, and I will introduce you shortly the Coriolis Air Samplers. So just for you to know, Bertin Technology can give you access to optimized solutions for life sciences. It's been more than 15 years that we have expertise in designing and manufacturing high quality lab equipment. So the one we will speak today is the Coriolis for air sampling, but we also have a free range of products. The Spécialiste, which is a new universal tissue homogenizer. We also have quite big range of bioreagent with ELISA kits, and we also have the InfOzyme, which is a smart cell imaging system. For the topics of today, Bertin has more than 10 years of wide cyclonic technology implementation, and we have developed different projects for more than 10 years. The first one was the Mona Lisa project. It was a European project for the pollen and allergen detection in the air. We also work with the French Army to develop for them a portable air sampler for airborne pathogen detection, such as anthrax. And for laboratory work, we developed the Coriolis Micro and the Coriolis Compact that we launched this year to collect all microorganisms in the air, such as bacteria, viruses, but also pollen or allergens. The Coriolis is used worldwide, as you can see here. It can be used in different projects on different topics, such as agriculture, but also for CBRN purpose, and in different countries, in the US, France, Asia. It's already used all around the world. The Coriolis Micro description, as you can see, the Coriolis is a small instrument, quite lightweight, with 2.8 kilograms. There is an autonomy of 1 hour of battery, but you can work with it up to 6 hours. And there is a battery which technology is NEMH. The airflow rate can be chosen between 100 to 300 liters per minute, and it collects, as we mentioned, any biological sample. It can be used indoor and outdoor, as it can be used on battery or on power supply. Regarding the technology, it's pretty simple. The thing is that you have a pre-filled sterile cone with adapted collection. So I heard that we have trouble with the slides. So is it better now? Do you see all the slide? Yeah. So I'm sorry. So I will show you. I will go back on the previous slide to see the Coriolis Micro, and then you have the Coriolis wave cyclone technology, which is explained. So you have a cone, which is pre-filled with adapted collection liquid. Then when you start the instrument, there is an airflow which apply, and you have a vortex which is formed inside the cone, and you have all the particles that are centrifuged with the collection liquid on the wall. It then allows to the particle to be collected and trapped inside the liquid. So when you stop the collection, all the bacteria or viruses are collected in the liquid. So to show you a more descriptive pictures, there is 4 parts in the Coriolis Micro. You have the body of the Coriolis, which is pretty simple, and you can see that the interface is really simple to be used. You have only one button to start the instrument, one to stop it, and arrow to navigate inside the menu. You have a cane of aspiration, you have an air intake, the section 3, and you have the cone filling with the liquid. We have now launched the new Coriolis Compact, which is a dry cyclonic technology. You can see that it's really lightweight. It's only 1.40 kilograms. It's really small. It's less than 25 centimeters high. The usage temperature is between 5 to 45 Celsius degrees. We have an autonomy up to 8 hours with a lithium-ion battery. The airflow rate is fixed at 50 liters per minute. It can also collect any biological sample, such as bacteria, viruses, pollen, allergens, or toxins. And it could be used indoor and outdoor, and we have a Bluetooth connection available. The dry cyclonic technology works the same way than the Coriolis Micro. So the cone is placed on the device. Then you aspirate the air, goes through the cone in the air intake, and it's working the same way. A vortex is created, and all the particles are collapsed inside the cone. And retained in the cone wall. At the end of the collection, you can recover the particles by rinsing the cone with the adapted liquid. The main advantages of the Coriolis Compact is that you collect more particles with up to 8 hours of straight sampling. So you just need to start the instrument and then wait for 8 hours. All the samples are compatible with rapid analysis methods such as qPCR. You don't need to concentrate your sample, as you can recover your sample in the desired volume, and it's easy to transport and can be put in strategic places or even on a drone if it's needed. Regarding the product description, you have the Coriolis body, which is in one piece. You have the air intake, which is a consumable as the cone, which is screwed on the air intake directly. Regarding the main advantages of the Coriolis, it's the fact that it's compatible with any biological analysis. So it's compatible with culture method if you want to check at bacteria. It's also compatible with immunological methods. What is in the trend right now is compatible with genetic methods for viruses, for example. So you can analyze your sample with qPCR, and it's also compatible with spectrometry method if you need it. And as you can imagine, the Coriolis can be used in any environmental monitoring, specifically for airborne viruses. So it's something that it can be used typically for the SARS-CoV-2 detection, so in food industry, but also in a veterinary environment, also in health and biomedical structures such as hospital, and of course in also office space if you want to control the decontamination. I will now give the microphone to Martin Serrano-Sanchez, who is our application engineer, and he will introduce you the tips and tricks to collect viruses in the air.
Hello, everybody. So this is Martin Serrano-Sanchez. So I will abord the subject of tips and tricks for the collection of viruses in the air. So when we are going to collect a virus, we have to think about sampling strategy, where we are going to place the device and how we are going to collect this sample, if there is a special media of what kind of media we can use, how we are going to handle this sample, storing, how are we going to process it, analysis, and also the decontamination of the device, because it has been in contact with virus. So we need to know what can we do to decontaminate our device. Okay, for the sampling strategy, right now we are talking about SARS-CoV-2. So our feedback and experience tells that it's better to make a run with an flow rate of 200 liters by minute. That is what we have seen and what has been described in literature. But anyway, there is not like a rule, and you can try other flow rates in order to collect virus. The duration of the sampling, well, it depends also of the conditions where you are sampling, the size of the room. There are so many factors. So here we are only pointing out some tips if you are new in using the Coriolis, and if you want to have an approach to better collect these molecules or virus. Okay, so we are suggesting a minimum sampling of 20-30 minutes. If you don't have the long time monitoring for the Coriolis Micro, you can make 2 or 3 runs straight. The device positioning. Well, the device positioning right now, as I said before, we are thinking about SARS-CoV-2. So it's interesting to collect the virus. Of course, if you are going to be close to a patient, you will have more chances to detect it. But people is now, wondering about how far the virus is going, and if there are risks to collect it in the places around the patients. So you can start from putting the device further distance away from the patient, and little by little move closer to the patient. You have to consider also that there is a natural movement of the air in the place where you are sampling. And so small particles or droplets generated by the patient are going to be also aspirated by this internal flow. So it's better to check in the site where you are sampling this kind of air movements and find best positioning for your device. The collection liquid that you can use, well, there are so many things that you can do. People is right now using PBS, and as we will see later in some examples, also DMEM media. This is also a suitable choice because it can keep in good shape viral particles. And the additive that it has, they work also as surfactants, so it allows good recovery of particles. When the Coriolis Micro is working, there is a natural evaporation of the liquid in the cone. So it's important to use this evaporation in your favor. As we know, the main technology or the main analysis that people is using to detect SARS-CoV-2 is PCR. So it's better to play with the volume, initial volume of your sampling, and work with something between 5 and 15 milliliters. So as you are going to be performing some runs, liquid is going to evaporate. And so if you have a smaller volume of liquid, it's going to be easier for you for concentrate your sample or either work directly with it. For the sample storage, after the collection of your sample, we suggest to transfer the liquid that you have collected into a proper tube, even to put it in the fridge, or if you desire to freeze it and keep it at -20 or -80. Also, depending on the final volume of your sample, maybe you will need to use a method for concentrate this sample. So many people use the Amicon or Centricon or similar to concentrate this final volume that can be around 5 or 10 milliliters. So they will have a concentrated sample at the end, and it's going to be easier for them to make RNA extraction or DNA extraction, depending on what you are looking for. For the Coriolis compact, you have an advantage because this new device collects in dry. So it's up to you to find the optimal buffer for recovering your sample. Mainly, any kind of lysis buffer that you add into the cone, vortex and scrapping a little bit the walls of the cone with them is going to be enough to recover a good quality RNA or DNA. For the samples analysis, as I said before, for the SARS-CoV-2 right now, people is more focused in PCR. So it's better to perform the RNA extraction as soon as possible. And when it's not possible, better to freeze their samples at -20 or -80 for later treatment. Concerning the decontamination of the device, of course, the best thing is to use H2O2 vapor, but we know that sometimes it's not possible in some facilities. So the best thing is like you can clean the outside of the device, the cane or the air intake with bleach water, ethanol 70%. Also, you can autoclave the cane and the air intake, not for the compact because it's not possible. And also for the inside of the instrument, if you don't have a device that is going to generate vapor of H2O2, you can put ethanol 70% in the cone, turn on the device and make a run or one or two runs of 10 minutes until the ethanol is going to be evaporated. So in this way, it will mainly deal with SARS-CoV-2, but I want to say that this kind of method won't be enough to kill, for example, spores, or you are not going to take out spores. I will present some case studies, and the focus of this is mainly to take a look at how people perform the air sampling for them. So we are going to visit the SARS-CoV-2 detection with the Coriolis Air Sampler, the norovirus detection in hospitals with Coriolis Micro 2, swine influenza virus detection in pig farms. And also we are going to show some results that we obtained with the Coriolis Compact. I will pass the microphone to my colleague, Sophie.
So Martin will introduce first the result that we obtained with a team at Imperial College in London. I would like just to thank them a lot, the authors, because they allow us to share the results and to present you the results this morning. Normally, Jisoo mentioned she will be here, but unfortunately she was not able to come. So we present ourselves the result, but I would like to thank them a lot for the collaboration we have today on this topic.
So I'm going to show you the detection of SARS-CoV-2 with the Coriolis Air Sampler. So the objective of this study was to evaluate the SARS-CoV-2's presence in air and also in surface during the peak of COVID pandemic in London. Samples were collected in air different sites, including 7 clinical areas and 1 public area at Northwestern London Hospital. In each area, 4 air samples were collected, and surface collected were also performed close to the place where air samples were made. As collection protocol, that this is really interesting, the sampling was performed using 5 milliliters of DMM, running the device at 100 liters by minute flow rate for 10 minutes. And from that, a small volume of the DMM was used to make the RNA extraction and later the RT-qPCR. So the sampling was performed just in the peak when they had patients present inside. What they obtained was around 50% of samples collected in surface were positive for SARS-CoV-2. Interesting, around 40% of the air samples were also positive. Anyway, as I said before, they were using only one fraction of collected air. They were really reducing to 1 cubic meter of air. So maybe a longer collection of air or bigger samples would match more with the surface samples result that they show. So even if they were able to collect virus, they detected the presence of SARS-CoV-2, they were unable to show that this virus was active. And the virus was also detected in public areas in the hospital. We are now going to take a look at norovirus detection in hospitals. So in this study, they were interested in norovirus because they are a risk for patients in hospitals, and these viruses are responsible for 50% of gastroenteritis outbreaks worldwide. So the goal was to look for the presence of norovirus G2 in aerosol during gastroenteritis outbreaks in the healthcare facilities. The strategy of sample, they positioned the device, the Coriolis Micro, 1 meter far away from each patient, in front of the patient's room, and at the nurse station. As parameters, they use 200 liters by minute, collection 10 minutes, and they use PBS as collection media. As we mentioned it before, this time people use filtration and concentration of the liquid sample with an Amicon device. And later, these samples were submitted to the RNA extraction and RT-qPCR. What they found was that in patient room, around 50% of the samples were positive. Same thing for the nurse stations, but also they were able to detect the virus in hallway and common areas. As I said before, air samplers were taken with Coriolis Micro at 200 liters by minute in 10 minutes. So as you see, this is a different approach, and it shows that sometimes concentrate the sample is also a good way to detect or what you are looking for. Now we will visit a case where Coriolis Micro was used in a veterinary field. So they were trying to understand how the swine influenza A virus is transmitted by aerosols. So the goal of this study was to determine the relationship between airborne swine influenza A virus and the number of infected pigs in farms. For this, they designed an experiment where they used pigs, 33 piglets, that were assigned to different rooms. Some of them had maternally derived antibodies. And anyway, some pigs were inoculated with virus, and they were put in contact with other pigs that were healthy, and they were separated by 30 centimeters apart from their pen. So infected pigs were in one side and mixed with uninfected, and non-infected were in other side, and the only way of transmission could be air. And they put the device between the two boxes. So they use 300 liters by minute during 10 minutes collecting in PBS. And they were collecting 3 times a week during 25 days after infection of the pigs. As before, they concentrate the liquid sample using Amicon device, and they perform RNA extraction and RT-qPCR. What they found was that from the 19, when they had the peak, all pigs were contaminated, what showed that the contamination was made by the air, and they were able to detect virus in air samples. Now I will show you a result that was obtained with the Coriolis compact. In this study, people were interested into the detection of the bacteriophage in dairy factories. So the goal, bacteriophage are around everywhere. So what people wanted to check if that there was an effective way to decontaminate certain areas in a dairy factory. For that, what people performed was a sampling strategy, collecting air samples before a decontamination treatment with ozone, just after treatment, 2 days after treatment with ozone. So the idea was really to evaluate if after the contamination process, you were able to touch the quantity of bacteriophage present in the site. For this, they use the Coriolis compact that has a flow rate of 50 liters by minute during 5 hours. And the sample was submitted to DNA extraction and later to simplex PCR for specific primers for Lactococcus. So what they found was that there was an impact of this treatment with ozone. Anyway, even if there was an impact, they were able to detect the presence of bacteriophage later. But today, we are not here to evaluate if the decontamination procedure works, because it works. The conditions when the samples were made were kind of hard, because those places were exposed to air entries everywhere. And anyway, the treatment seems to work for some of the phage, not for others. But the main thing is that Coriolis compact was able to collect bacteriophage that are more or less the size of the actual virus. Actually, this is a new device. We are waiting for results of people that are testing for collecting SARS-CoV-2 with Coriolis Compact. But as this technology has an advantage when you don't need to concentrate your sample, we are expecting to have soon some results concerning to SARS-CoV-2 to show that SARS-CoV-2 can be detected in the air by Coriolis Compact.
Thank you, Martin. So before the Q&A session, I just want to conclude. So as you understand, we have two different instruments, the Coriolis Micro and the Coriolis Compact, which are able to collect SARS-CoV-2 or any virus or bacteria in the air. With the Coriolis Micro, you have the advantages of having a high airflow rate platform, which allow you to do long-time monitoring, and you can have your sample directly in a liquid for molecular analysis. The Coriolis Compact Air Sampler is really easy to use and to transport on the field. So depending on where do you want to monitor the SARS-CoV-2 or any bacteria, viruses in the air, it can be really easy to use. And you have a dry sampling with no resuspension step before molecular analysis. It means that, as Martin mentioned, it's bring you your sample directly concentrated. So the main conclusion is that the Coriolis is an ideal instrument for airborne viruses detection. Just to complete, as we mentioned, we had already results with the Coriolis Micro for the SARS-CoV-2 detection. We are waiting other results coming from other countries, such as France and Belgium and Luxembourg, but we are also waiting results for the Coriolis Compact. So you will be updated as soon as we will have feedback from our user on this topic. So in the presentation, you will have my contact. So if you need any information, don't hesitate. We will now start the Q&A session. So just let me open the question box. We have a lot of questions, so I don't know if we will be able to go through all the questions, but we will. So. So we have one question. Do you have any data on how notable amount of PM suspended in the air affect sampling performance? As an example, poultry farms can be quite polluted and saturate sampling medium. So we have few feedbacks on that. Generally, we have no impact on the pollution with PM on the collection. I will let Martin adding some information on that.
Yes, there's different articles about the subject, but I don't think that PM, because mainly when people are interested in to collect in poultry, they had been performing PCR. So even if you have some PM, as you are going to perform the RNA or DNA extraction of your sample, they should not interfere in your analysis. So you use an aggressive buffer, or maybe you could, I don't know, make the extraction with trisol or similar solution. And so you will precipitate your RNA or DNA, and PM shouldn't be a problem. If you are talking about something that is bigger, maybe it could be easier to put a grid in the air intake in order to avoid big chunks of feeders or other stuff that could be there in your sample. But anyway, if you are performing DNA or RNA extraction, that should be fine.
So the next question is, for virus detection, what do you recommend, Coriolis Micro or Coriolis Compact?
Well, that is a hard question. If I check only what we have in the literature, I would say Coriolis Micro. But the thing is that the Coriolis Compact is a really new device that it has been out this year. So what I can say is, if I perform, I'm not talking about a particular organism. I put my Coriolis Micro and my Coriolis Compact in a place where I know that there is some biological particles, bacteria, virus, I don't know. I make a run of them for equivalent volume of air, because I don't need to concentrate the sample. When I make DNA or RNA extraction of both samples, I will find higher concentrations of DNA or RNA in the samples performed with Coriolis Compact. But as I said before, it's maybe because I don't have an intermediary step that forced me to concentrate my sample. So if I'm based only in the literature, I would set Coriolis Micro right now, but it's only because it has been a long time in the market and there are more studies performed with this device. But yeah, I think that Coriolis Compact is going to be effective. Anyway, as you know, bacteria and virus are never floating alone in the air. They all the time stick to bigger particles around the size of 2 or 3 micrometers. And this size of particles are pretty well collected by Coriolis Compact and Micro.
So we have one question regarding the flow rate. So how does the flow rate affect the collection efficiency?
Well, of course, it's going to affect, because you are going to kind of impact the particles in the liquid. But as I said before, concerning to virus, most of articles in the literature made already the experiment. And that's why we advise to collect virus at 200 liters by minute. That is what I can say. After that, flow rate is going to be affecting more the volume of air. But in Coriolis Micro, yes, it has an impact, because maybe the vortex that is formed inside, it's going to operate in a different way.
The next question is about the sampling time. So how long maximum can we sampling? So it depends of the instrument. With the Coriolis Micro, you have 10-minute sampling with the simple configuration, but you can go up to 6 hours of sampling with the long time monitoring option. And with the Coriolis Compact, you have an 8-hour sampling autonomy with the battery. But I will let Martin adding if there is something for the virus specifically.
Yes, as Sophie said, with the Coriolis Micro in the basic configuration, you will be able to make runs of 10 minutes maximum. So if you don't have the LTM, there is no problem. You can you go there and only click again in the play button, and you will go for a second run of 10 minutes. You only need to pay attention to the evaporation of the liquid, as in average, you are going to lose 5 milliliters every 10 minutes, depending on the temperature and the humidity of the place where you are sampling.
The next question is on the percentage of particle that is captured. So do you capture 100% of particles, or do you have efficiency depending on the size of the particle?
Yes, we are not able to collect 100% of the particles. For that, if you send us an email, we can send you back the curves that showing more or less the efficiency rate of each device. But I would say that for particles with Coriolis micro of 0.5 microns, yeah, 5 microns, I would say that is 50%.
So next question is about the collection liquid. Do we have any specification regarding the pH of the PBS which is used?
It was classical PBS, yeah. pH 7.
So we have more question about, yes, so the PDF slides will be sent after the after the end of the webinar. So you will receive an email with a link for the recording of the session and also the PDF slides. So no problem to receive everything. So we have several questions around the same topic regarding is it an answer with a qualitative signal or quantitative? So are we able to, with the PCR, to have something quantitative or only qualitative? Do you have any feedback on that, Martin?
Yeah, well, it depends of the kind of PCR, because you can make a relationship between the volume of air that you collected and the number of copies, but that is something that is more for the PCR specification of what you are looking for. Yeah, but you will have something that is quantitative.
We have a question. Related to another question we already had. So regarding the maximum duration of the sampling, and is there any saturation of the liquid or of the cone after sampling, depending of the Coriolis micro or Coriolis compact you are using?
Well, I have never seen something like saturation with Coriolis micro. There are people that have made runs from 1 hour. The only thing that maybe could affect is that if there are like big chunks of I don't know, feeders leaf that go inside the cone, that is something that you have to avoid if you are collecting or sampling in the outdoors. But no, concerning Coriolis compact, as is dry, I don't have the answer, but usually I think that the surface of the cone is quite important. So you really need to go so far to saturate that. Yeah, but it depends maybe if the environment where you are sampling is really full of particles. Yeah, maybe you could saturate the cone.
So we have other questions regarding the difference between the liquid sampling and the dry sampling. So the question is, what happens if all the liquid evaporates in the Coriolis micro, And another question, so can you confirm that with dry sampling, there is no liquid at the beginning and you just reconcentrate at the end with liquid?
Well, what happens if the liquid evaporates completely in Coriolis micro? So there will, I think that because you are going to make a kind of dry film, there will be still particles that were collected that are going to be sticked still in the cone. But if you let it run for quite long, maybe some of those particles are going to be escaping from the cone through the airflow. So that's why it's better to always have some liquid. Coriolis micro was not designed to keep the samples and collect in dry. It can work in dry, but as far as I know, this was made only for pollens.
Oh. So I'm going through the question. So what do you have any information regarding the impaction efficiency in relation to particle size and medium?
Yes, this information we can send you by email too. So the collection liquid that is mainly used is it has some triton or twin. So this is made because in this way you will reduce the superficial tension of the liquid and it will improve the collection of particles. As I said before, I think that for 5 micrometer, 0.5 micrometer particles, the efficiency of Coriolis micro is about 50%. And for the same size, I think that is around 80% for Coriolis compact.
So we have a specific question on SARS-CoV-2. So do you need to inactivate SARS-CoV-2 during the collection step with Coriolis micro? It is considered a dangerous pathogen and culture is not always allowed. So do you have any feedback? I can give the one.
Well, I mean, if you are trying to collect SARS-CoV-2, I think that you are already aware that it's dangerous, and I suppose that people is going to be taking the precaution that they are needed. So yes, once that you have made your collection, and if you are like protected, the best idea is to really clean up all the material that you used, and SARS-CoV-2 can be inactivated by using ethanol 70%. It has been shown there are papers around, and you can use any other solution, astringent solution that is going to take off wipe up the virus.
Yeah, so I can add that the feedback that we have from the team working on SARS-CoV-2 is that they followed all the recommendation to manipulate the sample recommended by the World Health Organization. It means that the technician or the engineer who is dealing with the sampling need to be wearing gloves, mask, glasses, and complete protection, as it mentioned in the WHO recommendation. And after that, you will need to manipulate the sample as a dangerous sample, of course. I think that we go all through the questions. So there are some questions that are in double. So I hope that you all have your answer, and we are almost at the end of the time which was allocated to us. So I would like first to thank all the participants, and I would like to add that we will send you the recording link in the presentation by email, so this week or next week. And if you have any question, please don't hesitate to send us all the question you have. And if you need specific document, don't hesitate to contact us to have all the information. I would like to thank you again, and to wish you a very good day, and I hope see you soon to speak about air sampling and virus detection. Thank you all, and have a good day.